adaptive cells, B and T cells, express B and T cell receptors on their surfaces that are able to identify myriads of specific pathogenic epitopes without hurting the self-epitopes or self-proteins. How does the human genome, with its limited spatial dimensions, allow for such vast epitopic possibilities? Well, this is done by VDJ recombination, that is also known as somatic recombination. I want you to know that the fact that we have the letter D within the parentheses is the fact that the heavy chain, and only heavy chain, has V, D, and J recombination. The light chains only have V and J recombination. So for now, to remember this fact, remember D for dense, D for heavy, and dense and heavy chain. VDJ recombination is a unique mechanism of genetic recombination that occurs only in developing lymphocytes and only during the earliest stages of T and B lymphocytic maturation. The process results in the highly diverse repertoire of antibodies, immunoglobulins, and T cell receptors that are found on B and T cells respectively. So, to summarize, VDJ recombination explains why we have so many antibodies, so many immunoglobulins, so many T cell receptors, and so many B cell receptors in our body. The process is a defining feature of the adaptive system, and its development was a key event in the evolution of the higher vertebrates. VDJ recombination occurs in the primary lymphoid organs, in a bone marrow for B cells and thymus for T cells, and in a nearly random fashion that rearranges the variable V joining J, and in some cases, with respect to the heavy chain, the diversity or the D gene segments. The process ultimately results in novel amino acid sequences in the antigen binding regions of immunoglobulins and T cell receptors that would allow for the recognition of antigens from nearly all pathogens, including bacteria, viruses, parasites, and worms, as well as altered self cells, as it is seen with altered cells or altered self proteins with regard to cancer cells. The recognition can also be allergic in nature. For instance, it can be a response to pollen or to other allergens, or maybe autoreactive, and this may lead to autoimmunity, as we're going to discuss down the road. Let's look at the antibody structure and gene segments that encode immunoglobulin chains. And first, let's see how we can make so many antibodies, immunoglobulins, and how the light chains and how the V and J recombination allows so many different light chains to be produced. The light chains are either encoded by the kappa or lambda light chains. However, each B cell in the lymphoid tissues possess either the kappa or lambda light chains, and never both together. So for now, the light chains are either kappa or lambda. The gene segment for kappa chain, as you see in the diagram, is on chromosome 2, whereas the gene segment for lambda is on chromosome 22. The most abundant form of the light chains in humans is the kappa chain. For instance, for every 3 kappa chains, we have only 1 lambda chain. So 75% of the light chains in human beings are kappa light chains. The variable domain of the light chains is encoded by random recombination of V and J exons. There are about 100 variable V segments for either kappa or lambda chains. There are 5 J segments for kappa and 6 for lambda chains, as again you see in the table. There is only 1 constant segment for the kappa and there are 6 for the lambda chain. The first step of recombination for light chains involves joining randomly the V and J chains to make a VJ complex before adding the constant or the C segment into that complex. And this happens during primary transcription. And as you see in the diagram below, I'm showing the V segment in red. As I said, there are 100 of those segments. However, in this diagram, I'm showing only 4 of them. With regard to J segment in green, I'm showing all 5. And with regard to C segment, I'm showing only 1. So this should be a representative of a kappa chain, the diagram that you see below. With regard to the heavy chains, first and foremost, in contrast to light chains, there is only 1 heavy chain, and the segments encoding for it are all on the chromosome 14. In contrast to light chains, the heavy chain chromosomes also include the D or diversity segment. The variable domain of heavy chain is encoded by random recombination of 70 to 250 variable, 30 diversity, 6 joining, and 9 constant exons. Of course, the numbers are variable, and some authorities believe that for the variable we have a lot more than 250. But again, to make it easy for you, the test makers on the USMLE and COMLEX are not interested in these type of numbers. These are just a quick review for you to have heard of it at least once and to orient you. When I get to the concepts that are more relevant to your exam, I will dwell on them more and I will explain them more for you. In the developing B cells, the first and foremost recombination event is between 1D and 1J gene segment of the heavy chain locus, as you see in the diagram. Any DNA segment between these 2 gene segments is then deleted. The ones that we don't need, we delete. This DJ recombination is then followed by joining a 1V gene segment from the region upstream of the newly formed DJ complex, forming a rearranged VDJ gene segment. All other gene segments between V and D segment are then deleted from the genome. Now we have a heavy chain with a rearranged and unique VDJ recombination. B cell receptors are membrane-bound immunoglobulins composed of 2 heavy, 2 light chains. The 2 light chains, as we said earlier, are either 2 kappa or 2 lambda chains, but not both. Remember this fact, not both. So your immunoglobulins for the light chains, either they have kappa, at least for most part, because 3/4 are kappa and 1/4 of them are lambda, and not both. There are almost 2 times more B cell receptors with kappa light chains than those with lambda chains. What is the reason for this finding? Well, I'm going to be referring to this diagram for you, and at the bottom of the diagram I'm showing the spatial location of V, D, J, and then mu, delta, gamma, epsilon, and alpha segments sequentially from 5' location on the chromosome to 3' location. During differentiation of B-cell receptors in bone marrow, the first step is development of heavy chains and then light chains. I want to orient you to these terminologies that quite often confuse medical students, and again, from a standpoint of the utility on USMLE and COMLEX, they are not as important. The primitive B cells are called pro-B cells, pre-B cells, and mature B cells. So sequentially from pro to pre to mature. So just lock in that information. All pro-B cells that are unable to develop a heavy chain will receive a death signal, apoptosis. Those that develop a heavy chain are called a pre-B cell, and they can proceed to develop a kappa light chain. So, what is the characteristic of a pro-B cell? It has a heavy chain. Now, that heavy chain, of course, this is the one that will determine the isotype of the antibody. Is it IgM? Is it IgG? Is it IgE? As we're going to see later. They first get the chance to make a kappa chain. If they fail to make a kappa, they may make a lambda chain instead. And if they cannot develop either of the two, they receive a death signal. Given that the formation of the kappa chains precedes lambda chains, a higher number of naturally occurring immunoglobulins, about 65%, possess a kappa light chain. Note that D and J segments of the heavy chains are on chromosome 14, as you see in the lower diagram. I'm showing the chromosome 14. And they will first undergo recombination to make the DJ segment. That's the first recombination between D and J, downstream. So it means that closer to the 3' location of the chromosome. Also note that the mu segment or exon of the constant region of the heavy chain is the closest segment to the J segment and it stays bound to the recombined DJ segment. This explains why, as we're going to see later, the primordial, or the first, if you wish, immunoglobulin that is produced in the body is IgM. So for now, lock in this information. IgM is the first immunoglobulin, and M also for the mother, M for the mom, and that helps you to remember that this is the very first immunoglobulin that we make. And again, it's located upstream from 3' location. It's closer to the 5' location and closer to the J segment. After recombination of V segment and combined DJ segment in conjunction with a constant mu segment of the heavy chain and splicing of the introns, means the segments that we don't need anymore on the heavy chain, now we make our messenger RNA. And then it is translated to a heavy chain protein. At this point, the pro-B cell is officially a pre-B cell. Now let's go back and looking at the light chains. I want you to know that the first step in development of light chains include recombination of the V and J on the kappa segment of chromosome 2. Also note that light chains do not have a D segment, as I said before. So if B cell fails to develop couple light chains, then it attempts to make a lambda chain from gene segments on chromosome 22. And if it fails to make a lambda, then it receives a death signal. Note that after the B cell becomes productive, means that it can make an immunoglobulin, then it has the challenge of increasing the diversity of the variable binding sites. So this is just the beginning that we have made the heavy and light chain immunoglobulin. We have to tune it up and work on it later to increase even the diversity of the variable binding sites even further. Just to note, T cell genes also have similar segments on two chains, alpha and beta chains. The V segment of T cell beta chain has 52 segments, the D has 2, and the J has 13 segments. This is a second tier fact for your exam. Again, you have heard of it once to just orient you that there are some similarities and differences between T cells and T cell receptors and immunoglobulins. Back to that case scenario, I'm going back to the concept of serum protein electrophoresis, and in this diagram I'm showing the serum electrophoresis of a patient on top, and I want you to compare it with the normal finding at the bottom. I have 5 patients in here, and I want you to tell me the picture on top belongs to which of the following patients. As you can see in the diagram, we have a flat amplitude in the gamma globulin position, position number 5. So this patient does not have gamma globulin. Among the given options, the best answer is going to be a 12-year-old boy with a history of frequent viral bacterial infections and markedly reduced levels of CD19 lymphocytes. Means those are the B cells, of course, CD19, and B cells are making plasma cells are making immunoglobulins. Therefore, this child is unable to make immunoglobulins, therefore he gets tons of viral and bacterial infections. Again, to orient you, the cells with CD19 protein on their surface are B cells or B lymphocytes. B cells are responsible for antibody formation either directly or indirectly by differentiating into plasma cells. In the cited serum protein electrophoresis, the protein spikes 1, 2, 3, 4, and 5. They represent albumin, alpha-1 globulin, alpha-2 globulin, beta globulin, and gamma globulin, which is flattened out in this diagram. As is shown, the 5th wave that normally represents the gamma globulins is missing. It's therefore very likely that the 12-month boy may have X-linked, and again, that's why we mentioned the boy, X-linked gamma globulinemia, or XLA, that is also known as Bruton type A gamma globulinemia. XLA is often suspected in a young child with a history of recurrent infections, and especially respiratory infections from 6 months of age onward. The reason is that the mother's IgGs that are transferred through the placenta can safeguard these children until 6 months of age. The diagnosis becomes more apparent when blood tests show the complete lack of circulating B cells as determined by B cell markers such as CD19 or CD20, in addition to low levels of all antibody classes, IgG, IgA, IgM, IgE, and IgD. Quite often to confirm the diagnosis, a Western blot test may be ordered to determine the expression of BTK protein and BTK mutation. This is the protein which we believe is mutated in XLA. Note that BTK enzyme plays an essential role in maturation of B cells in the bone marrow, and when mutated, the immature pro-B lymphocytes are unable to develop into pre-B lymphocytes. As a result, neither mature B cells nor plasma cells, and in turn no gamma globulin, will be present in the serum of these boys. I'm sure that you are interested to know a little about the alternative options, who those patients are, and what type of characteristic serum electrophoresis findings you see in them. For the first option, option A, a 65-year-old man with a 9-month history of backache and hypercalcemia, whose skull X-ray is significant for lytic punched bony lesions. That guy is a patient who has multiple myeloma. So the 65-year-old patient has multiple myeloma, and the case scenario in red is a classic symptoms age-wise, chronic backache. This is a common finding. Now these patients actually should have had a gamma spike, a narrow, tall spike, and that tall spike is either due to IgG or IgA immunoglobulins. So if the picture was like this that I'm showing, then you might have picked up option A. With regard to the next option, option C, a 60-year-old man with a history of diabetes type 2 who has bilateral dependent edema, foamy urine, and a recent history of deep venous thrombosis of the calf veins. So what type of finding you would have liked to see in this patient? Just to know, patients most likely with foamy urine and tells us that the patient must have gotten to the point of later stages of nephrotic syndrome, because diabetic nephropathy is one of the most dreaded consequences of diabetes mellitus. So that foamy urine is an indication. And also, of course, the bilateral edema also is another major association of nephrotic syndrome. Now, the first prominent serological finding in these patients is loss of serum albumin that is presented on the electrophoresis by a flat spike at the position number 1. So instead of this tall spike, you should have seen a flat effect for essentially position number 1, albumin, because we lose all the albumin or most of it into urine as a result of nephrotic syndrome. So for these patients with nephrotic syndrome, you don't see that spike. If so, to summarize, if you do electrophoresis for patients who have nephrotic syndrome, you will not see that spike in the position of the albumin. That one flattens out. The next patient, a 66-year-old man who is diagnosed by a disease characterized by somatic mutation in MYD88 protein, and as a result has uncontrolled clonal proliferation of terminally differentiated B lymphocytes. I covered this a little earlier for you, and I'm just drilling you on it. This patient most likely has Waldenstrom macroglobulinemia, that is also known as lymphoplasmacytic lymphoma. The serum electrophoresis in patients at the position 5, the same way that I'm showing, they should show a tall spike, but this time this is due to uncontrolled increase in the level of the IgM. You have to contrast this with multiple myeloma, that red spike in multiple myeloma was due to IgG and IgA. In Waldenstrom, this is due to IgM. Finally, A 25-year-old overweight woman who is diagnosed with familial hypercholesterolemia. Patients with severe familial hypercholesterolemia present with high levels of low-density lipoproteins, LDLs, and early symptoms of coronary heart disease. I want you to know that transferrin and LDL are the two major serum proteins that normally occupy the beta globulin position, number 4, in the serum electrophoresis. And patients with familial hypercholesterolemia are expected to have a very tall or peaked beta globulin in the position number 4. So the electrophoresis of this woman that at that early age presents with coronary heart disease and a high level of the LDLs should show a peaked sort of globulin levels at the beta globulin position, as you see in this diagram. Let's change the subject a little. The innate immune cells, such as macrophages and dendritic cells, are the first-line defense against pathogens, and they are the ones that relay the information and present the antigens to the adaptive cells, the B and T cells, in the lymph nodes. What are the two main regulatory molecules that facilitate interaction of innate and adaptive B and T cells? As you can tell, what I'm heading for is to let you know how the cells within the innate system and within the adaptive system and between the innate and adaptive system, they communicate with each other. So what are the two main regulatory molecules that facilitate interaction of innate and adaptive B and T cells? I'm heading for the concept of major histocompatibility complex, and there are two classes, class 1 and 2. Just to know, MHC is also known as human leukocytic antigen. Again, we have class 1 and 2 of these leukocytic antigens. These proteins are encoded by human leukocytic antigen gene complexes that are highly polymorphic genes with multiple alleles located on chromosome 6p21. So let's take a little time and talk about major histocompatibility complexes. The HLA molecules are more antibody-like than antigen-like. What is the reason for calling them antigens? Well, this is due to their original historical discovery in conjunction with blood and organ transplantation. Human leukocytic antigens identification began as a list of antigens involved in transplant rejection historically. HLAs are not typical antigens like those found on the surface of infectious agents. These are alloantigens and cause immune response to foreign alloantigens from members of the same species. More specifically, HLAs are involved in alloimmunity. Alloimmunity is caused by difference between products of highly polymorphic genes, primarily genes of MHC complex of the donor and graft recipient. These products are recognized by T lymphocytes and other mononuclear leukocytes which infiltrate the graft and damage it. I will be talking about organ transplantation a little down the road, but we are just orienting ourselves at this point. So I'm going to be touch basing with these concepts from a more clinical angle a little later for you. So let's compare and contrast the MHC 1 and 2 with each other. They are both similar because they are both glycoproteins, both encoded by tightly held clusters of genes on chromosome 6. The MHC genes also encode for complements C4, C2, and complement B factor, TN-alpha, tumor necrosis factor alpha, and some cytochromes such as cytochrome P21. Both MHC, actually I have a note here for you in case you have been interested about the B factor. Complement factor B is part of the alternate pathway of the complement system and is cleaved by factor D into two fragments, fragments A and B, and ultimately it causes formation of the complement 3 or activation of complement 3. And we'll cover the complement stories for you a little later down the road. But for now, I want you to know that in addition to the MHC1 and 2 genes, we have also genes for complements and some cytochromes on chromosome 6, or in the same segment that encodes for MHC1 and 2. On chromosome 6, we also see encoding genes for these complements. So both MHC clusters are involved in graft rejection and clearance of exogenous antigens. Both have a surface group that binds a peptide for presenting T cells that can only recognize antigens in conjunction with the MHC molecules. So for now, T cells can only recognize antigens if they are presented on MHC molecules. Now the contrast between the two is as follows. MHC1 is present on all nucleated cells, that is all cells except red blood cells, as you can tell. MHC2 is present only on the surface of antigen-presenting cells, APCs. Who are they? Remember these. There are 3 APCs: macrophages, dendritic cells, and B cells. So B cells can also act as an antigen-presenting cell. MHC1 interacts with cytotoxic T cells, whereas MHC2 interacts with T helper cells. MHC1 binds T cell receptors, TCR, and CD8 molecules on cytotoxic T cells. I'll be talking more about these later. It presents peptides that are smaller in size, 8 to 9 amino acid long. Whereas MHC2 binds T cell receptors and CD4 adhesion molecules on T helper cells. It presents peptides that are 13 to 18 amino acid long. MHC1 is encoded by HLA-A, HLA-B, and HLA-C genes on chromosome 6. I'm going to show you the diagram to you shortly. Whereas MHC2 is encoded by HLA-D genes. Those are DP, DQ, and DR genes. MHC1 consists of a long alpha and a short beta chain or beta globulin. And just to know, only alpha chain is a membrane spanning chain. Means that it passes from outside into the inside of the cells. MHC2 consists of 2 similar length alpha and beta chains. Both chains are membrane spanning. MHC1 binds endogenous and intracellular antigens such as viruses that invade these cells and use cellular energy sources for making new viral particles. To give you a little mnemonic and understanding about this concept, I just said that the MHC1 presents peptides that are shorter, 8 to 9 amino acids. and you see it on all nucleated cells. So these MHC1s, as we're going to see later, they also present myriads of self-proteins that can be small proteins, and they are more sort of refined in their activity, and they can recognize small chunks of proteins only a few amino acids long. And of course, the viruses also are smaller in size. So this will help you to understand why MHC1 presents smaller size peptides compared to MHC2, which deals with exogenous or extracellular bacteria, for instance, or germs that are relatively bigger in size. MHC1 binds endogenous and intracellular antigens such as viruses that invade the cells and use cellular energy sources for making new viral particles. Whereas MHC2 binds exogenous and extracellular foreign antigens such as bacteria. With regard to MHC1, when endogenous proteins get degraded, the peptide fragments of those are transported to the endoplasmic reticulum by binding to MHC1 proteins before being transported via the Golgi apparatus to the cell surface. With regard to MHC2, upon encountering the pathogenic proteins, the APC cells degrade them into peptide fragments and retain them within their endosomes before transporting them to their surfaces and binding to MHC2 proteins. MHC1 also presents surface proteins for monitoring by the natural killer cells as part of a routine surveillance system that identifies and destroys cells with unfamiliar surface peptides. And cells with abundance or lack of certain peptides or presence of malignancies that will affect the surface MHC1 expression. With regard to MHC2, binding of helper T cells to B cell MHC2 antigen stimulates development of a clone of antibody producing cells against the current and future antigenic material. Well, this caricature picture and mnemonic helps you to distinguish the key characteristics of MHC1 and 2. First and foremost, MHC1 is a smaller number, and as such, the MHC1 can present proteins that have only 8 to 9 amino acids. In other words, it can load smaller proteins, whereas MHC2 can load bigger proteins, proteins that they have 13 to 18 amino acids, or antigens that they have 13 to 18 amino acids. Also, MHC1 has only one chain, the alpha chain, or one leg, if you wish, that passes through the cell membrane. So it has one transmembrane chain, or for the sake of mnemonic, it has only one leg. Whereas the MHC2, in the diagrams that you see in the literature, it has two legs that are transmembrane, an alpha and a beta chain that pass through the cell membrane. The other mnemonic to remember, another association, is that MHC1 is associated with HLAs that have only one letter, such as HLA-A, B, and C. Whereas MHC2 is associated with HLAs that have two letters, such as HLA-DP, HLA-DQ, and HLA-DR. Also, MHC2 presents the antigens to Th4 lymphocytes, whereas the MHC1 presents the antigens to CD8 lymphocytes. So MHC2 binds to T cells with CD4 markers on them, whereas the MHC1 binds to T cells with CD8 markers. Mnemonic 1 multiplied by 8 gives you 8, 2 multiplied by 4 gives you 8. So what is the cell that this MHC dude is leaning on? Well, of course, this MHC has two legs, and two-legged MHCs are only seen on antigen-presenting cells. But this most likely is a B cell. Why most likely a B cell? Because it has a very big nucleus. But I want you to know, potentially, this might have been a macrophage or dendritic cells as well. Of course, the dendritic cells, they have those dendrites on them, those hair-like projections on their surfaces, and there is no hair-like projections on this cell. But anyhow, this is most likely a B cell. And remember, earlier I said that B cells can also act as antigen-presenting cells. Let's look at the genetic organization of human leukocytic antigen. I have a good diagram here, and you see that the HLA region is part of the short arm of the chromosome 6, and if I enlarge that segment, the HLA region, you're going to see within it the class 2 region that includes HLA-DP, HLA-DQ, and HLA-DR genes. Then we have the class 3 region that includes those complements that we talked earlier about, complement 4, complement 2, cytochrome P21, and also it has genes for tumor necrosis factor alpha and beta. And finally, we have class region 1 that, as you recall, it has 1 letters in their names. Those are HLA B, C, and A. HLA complex is located on the short arm of the chromosome 6, as we said. The complex exhibits a high degree of polymorphism, and HLA loci is extremely variable. And as we're going to see later, this plays an important role in autoimmune diseases. Over 2,000 alleles are identified for HLA-A, 100 for HLA-B, and 40 for HLA-C. Interestingly, the number of these alleles are progressively increasing. So you may not be surprised if you read the literature a few years down the road, you may see a much higher number for HLA-B or HLA-C or HLA-A. Almost all individuals are heterozygous for most MHC foci, and practically no two individuals have 100% identical HLA alleles. HLA complex, in addition to encoding for the 6 classic transplantation HLA genes, encodes many more proteins that play roles in regulation of immune system, as well as some other fundamental molecular and cellular processes. For instance, tumor necrosis alpha and complements 2 and 4. Given that the surface HLA molecules present antigens to adaptive and innate cells, this polymorphism would allow the hosts to mount a much wider response to a much wider number of pathogens. The HLA segment of human genome is associated with more than 100 different diseases. And again, the numbers are increasing as time goes on. These diseases include, but they are not limited to, diabetes, rheumatoid arthritis, psoriasis, asthma, celiac disease, Graves' disease, ankylosing spondylitis, and systemic lupus erythematosus. Let's compare and contrast the way that MHC1 and MHC2 present the antigens or proteins. MHC1 presents endogenous antigens, such as viral particles or self-proteins. In contrast, MHC2 presents exogenous antigens, such as bacterial particles. The cytosolic antigen for MHC1 is degraded by proteasomes, whereas the exogenous antigen of MHC2 is internalized first by phagocytosis and endocytosis, and then degraded into smaller particles within the vesicles. MHC1 particles are transported to rough endoplasmic reticulum, and in there they are loaded onto MHC1 molecules. Whereas the MHC2 molecule is synthesized in rough endoplasmic reticulum and independently is transported to vesicles that contain degraded pathogens. With regard to MHC1, the MHC peptide complex is transported via Golgi apparatus to the cell surface. With regard to the MHC2, the antigens are loaded on the MHC2 and then they are presented on the surface of the APC cells. I will show the diagram to you shortly. Well, here is the diagram. We are going to be talking about the MHC2 and antigen presentation. Of course, you see two legs here, two legs all over. These are MHC2s. And of course, I'm showing the external pathogen that must be phagocytized. So let's go through these phases together. Antigen presenting cells, such as dendritic cells, phagocytize the exogenous pathogens such as bacteria. This is event number 4. And degrade them into fragments by enzymatic digestion within their acidic intracellular endosomes. This is event number 5. And then these vesicles are infused. These vesicles are fused with Golgi apparatus. This is event number 3. Now, from a different angle, the MHC2 molecules are encoded for in the nucleus of the APC cells, and they are synthesized in the cytoplasm, transported to rough endoplasmic reticulum, and then into the vesicles. They originate, at least the genomic code for them is in the nucleus, and then they enter into rough endoplasmic reticulum. But this is important. I want you to lock in this name. They are transported from the rough endoplasmic reticulum into vesicles by the invariant chain. That's the event number 2. So I have to explain the invariant chain for you. The invariant chain, or CD74, is a protein required for MHC2 transport and shaping of its binding groove. In the rough endoplasmic reticulum, it causes association and stabilization of the alpha and beta chains of MHC2. Without this chain, the alpha and beta proteins of the MHC2 will not associate. So this is the important issue about the invariant chain. So far we brought the MHC2 molecule or protein into the vesicles. Now that we have brought the MHC2 into the Golgi apparatus, the next step is fusion of the acidic endosomes, that they have the bacterial particles, with the Golgi apparatus, and the antigen is now loaded on the surface of the MHC2, on the available antigen binding groove or site on the MHC2 molecules. And finally, the next step, the antigen MHC2 complex, is now presented on the surface of the dendritic cells or antigen presenting cells. Note that similar steps may also be performed in other APC cells such as macrophages and B lymphocytes. This diagram shows antigen processing and presentation on the MHC1 cells. Remember, every nucleated cell has MHC1 on it. Remember that the MHC1 presents endogenous particles. So endogenous or cytosolic antigens such as viral particles, and here I'm showing a viral particle, different parts of the viral particles have different colors, are degraded into peptide fragments by proteasomes, and I'm showing the proteasome in blue, and converted onto peptide segments, and I'm showing different peptide segments here, within nucleated cells of the body. The peptide segments are then transported from cytosol into the lumen of the rough endoplasmic reticulum by TAP. TAP stands for Transport Associated with Antigen Processing. Transport associated with antigen processing. This molecule or protein is essential for delivery of peptides into the lumen of rough endoplasmic reticulum, where they are loaded onto the MHC1 molecules. So we can get these peptides, and now we load them on MHC1. And of course, the MHC1 is encoded in the nucleus of every nucleated cell and then is synthesized in rough endoplasmic reticulum. The MHC peptide complex, this is the one, is transported now via the Golgi apparatus, via secretory vesicles onto the surface of the nucleated cells. Note that the surface MHC1 antigen complex will present the antigen, this time to CD8 positive or cytotoxic T cells. Again, note that proteasomes and lysosomes are two major intracellular devices that help to degrade proteins. Lysosomes deal with extracellular proteins and proteasomes with endogenous proteins. So we see lysosomes in association with MHC2, and we see proteasomes in association with MHC1.